There are two different users of PanelApp, public or anonymous, and expert reviewers. To maximise engagement with the clinical and research community, we have made PanelApp publicly available. As a public user, you can view and download each gene panel. You can also view evaluations and comments made by expert reviewers. As a public user, you will not be able to evaluate genes or add new genes to panels. To find out how to become a PanelApp reviewer, see our videos on reviewing panels. The homepage tabs contain background information, guidance for reviewers, news and updates, instructions and how-to, frequently asked questions, contact details, and source information. From the homepage, you can choose whether to search PanelApp by gene or by panel. To view the list of gene panels available on PanelApp, click the Panels button on the top menu. All available panels will be shown. The list can be sorted by name, the number of genes that have been reviewed, or the number of reviewers. The list can also be filtered by searching for keywords, such as cardiovascular, to view all of the cardiovascular disorders, or by version number. The number to the right of the search box will tell you how many panels are in your filtered search. To view the gene panel, click on the disorder name. On the gene panel page, you can read the Genomics England eligibility statement criteria for that rare disease in the description box. Eligibility statements for all the rare diseases can also be found on the Genomics England website. Scroll down to view a list of experts that have reviewed the panel and their affiliations. Scroll to view the list of genes in the panel, how many genes have been reviewed, and how many genes are ranked green, diagnostic grade. Each gene in a panel has been given an initial level of confidence based on the number of the 4 sources, Emory, Illumina, UKGTN and Radboud, that the gene was collected from, as indicated by a traffic light system. Green indicates the highest level of confidence, a gene from 3 or 4 of the sources. Amber indicates intermediate confidence, a gene from 2 sources. And red shows a lowest level of confidence, where the gene was selected from 1 of the 4 sources or from an expert list. Based on the guidelines available on the PanelApp homepage, we're asking experts to review this rating and provide their evaluation of whether the gene should be on the green list, a high level of evidence for an association with the disease, or the red list, a low level of evidence or not possible to report clinically. For each gene, you can see the number of reviews and what the ratings were. So for example, for CD79A, there are 2 experts who think this should be on the green list and 1 that thinks it should remain on the red list. Also listed is the mode of inheritance, if available, the sources of evidence the gene was found in, and the phenotypes captured from the sources, which includes both phenotypes and OMIM disease names and identifiers. On the gene page, you can view the reviews and comments made by each expert. Click on the Details tab to view more information about the gene, links to other databases such as Ensemble, OMIM and Gene to Phenotype, and other panels in PanelApp that contain this gene. Use the arrows or click on a gene symbol to scroll through the genes on the panel. The grey genes at the bottom of the panel are those added by reviewers during their evaluation. These will be assessed by a curator and rated red or green according to expert review and our curation guidelines. Click on the panel title to return to the whole panel view. A selection of downloads is available at the bottom of the page, with the option of selecting a previous version of the panel or just the green, diagnostic grade genes. As you can see, as a public user, you cannot give reviews for genes or add genes to the panel. To do this, you will need to register as a reviewer on the login page. From the panel search page, you can also compare the genes set on 2 different panels. Choose 2 panels from the drop-down list. The comparison will show you the rating of each gene, which genes are shared between panels, and which genes are found on only one of the 2 panels. Scroll down to see the full list. Alternatively, to search for genes within PanelApp, click on the Genes option from the top menu. Scroll down to locate your gene of interest or type a gene symbol into the search box. PanelApp will display all the panels that contain that gene and show the rating, green, red, amber or grey, of the gene on each panel, and additional information associated with that gene and disorder, including mode of inheritance and phenotypes. Clicking on a gene will take you to the gene page for that panel. You can retrieve a list of all the panels that gene features on from this page. Further searches can be performed using the PanelApp API. For more information on PanelApp web services, see our web services video.